Thanks very much, and thank you for coming out tonight. I would like to talk to you about mitochondria. It's something I've been interested in since my PhD, and particularly my postdoc, which was at NIEHS, and how they are affected by environmental exposures. I'd like to draw your attention to the beautiful picture up here. This is a picture of a human cell taken by one of my former PhD students, Amanda Bess. And I'd like to ask, how many of you know what the blue thing in the middle is? Just shout it out if you know. Nucleus, that's the nuclear DNA. What about the red stuff? Spaghetti? Sorry? Mitochondria. That is the mitochondrial network. Notice that it's a network, not a bunch of little kidney beans things. How about the green, yellow stuff, the little dots in the red? Okay, someone said mitochondrial DNA. That is absolutely correct. Great. Let me say a little bit more about mitochondria and then mitochondrial DNA. So you've probably heard of mitochondria. They're frequently referred to as the cellular powerhouses. They generate a molecule called ATP, which is shown here. And this is the cellular energy currency. This is what allows our cells to do work. It's not a trivial task. Our mitochondria produce our body weight in ATP every single day and turn it over. It's an amazing amount of ATP made. That's not all that mitochondria do. They also function in immune function. They also function in making heme for hemoglobin. They may even underlie the meaning of life. I won't make that claim. You can talk to my mom. She's here. She's a theologian, so she's the authority there. In addition, mitochondria, as pointed out, have DNA. It's much, much smaller than the nuclear genome. It's about 200,000 times smaller. But there are many of them in the cell. Despite the small size, it's quite important, and we know that because there are a lot of diseases caused by mutations in that genome, in that tiny little genome spread out throughout the cell. From a toxicologist perspective, what was fascinating to me is that some of the repair mechanisms that allow DNA to be fixed in the nuclear genome don't exist in mitochondria. And so that raised for me the question of if you have an environmental exposure that damages that genome, what happens? And in addition, what happens to that damaged genome? Where does it go if you can't fix it? So we've done a lot of experiments to try to address this question, and environmental exposures that cause damage alter mitochondrial function, alter the levels of the ATP I told you about. At the level of the organism, I don't work with voles, I don't work with rats or mice even. I work with worms. This is a picture of the worm I work, well, this is the head of the worm I work with. This is the brain. We're visualizing here one population of neurons in its head, visualized with green fluorescent protein that's produced only in those cells. And in normal, unexposed worms, you see nice, beautiful cell bodies and nice, clean extensions. And when exposed to mitochondrial DNA damage, what we see is blebbing and thinning of these neurons. These are the same neurons that degenerate in Parkinson's disease. So we think there are important impacts of damage to mitochondria. I still haven't told you what happens to those damaged genomes, and what our data suggests, what our data suggests is that it is recycled. So hopefully it will stay there. So in this picture, the mitochondria are the green. Again, a network, as we've already talked about. What I haven't told you is that is an active network. It fuses, it divides. Sometimes little pieces come off and are sent to lysosomes, as shown in red here. These are the cellular recycling centers, and they break up the mitochondria. And because there are multiple mitochondria in DNA, they can simply be replaced. So this allows a response to that kind of damage. Now, this evening's talk and focus is on gene-environment interactions. I haven't said much about that yet. This is best understood in the context of drugs. There are some very clear examples there. One example is the antibiotic gentamicin. This is safely used by the great majority of the population, but folks who have a specific mitochondrial DNA mutation can go deaf from using this antibiotic. I tried to find an image that would illustrate this idea, and I failed. But I did find an advertisement for a lawyer who will help you sue the manufacturer of gentamicin if you suffer from this problem. So it's a real issue. It affects a significant number of people. What about pollutants? So pollutants are not as well studied toxicologically as drugs. There are many more of them, and our exposure is much less well understood. Susan will fix this for us, but for the time being, it's not as well understood as we'd like. So what we wanted to know is if we take these same worms in which we see neurodegeneration, but now we look at a subset of the worms in which the mitochondria don't fuse because of a genetic deficit, will they be worse off? And we did this experiment, and the worms in which the mitochondria fuse fine grow up, and we can look at neurodegeneration. We couldn't actually do the experiment in worms in which mitochondria don't fuse. They got stuck at the first larval stage and died. Maybe that's why they've gone away. They actually did die. So we actually couldn't do the experiment. We don't really know yet why that occurs. It's something we're continuing to study, so stay tuned on that one. And I'm not sure if I can get to the last slide, but if I can't, that was a thank you to my lab for doing the work. Oh, well, that's the dead worms, but there's also supposed to be a thank you to my lab, a thank you to all of you for your attention, and your tax dollars which support this work. Thank you.